Hey guys, welcome to another video, the first video of its kind. And yeah, this is me trying to pass some knowledge, some things I've learned in biochemistry. So I know we have this exam coming tomorrow. So if all goes well, you see this video tonight and hopefully you learn something from it. Okay, so let's start with what we talk about in carbohydrates. So I'm just going to do a brief overview because if we talk too long, this is going to take forever. Okay, oh, I have a laser pointer. Oh, that's nice. I didn't know I could do that. Okay. Anyway, so let's go on. So what we have is carbohydrates, and it's a lot of material for carbohydrates, but I think very soon you will understand why it's a lot. Yeah. So carbohydrates are known as saccharides. We know this, the most abundant molecules. So I'm not going to really talk about this stuff because you actually know all of this and you can actually read this. Polymeric carbohydrates are also found in cell walls and in protective coatings on organisms. So they can ask us questions about where are they found and stuff like that. So we go on to carbohydrates can be described by number of the monomeric units they contain. So in other words, a monomer of carbon is like the smallest subunit. So for example, you have three carbon atoms together. That can be a monomer. Glucose has six. Fructose. What else is there? As we go down, we start seeing that monomers combine to form polymers. So monosaccharides are the smallest units of carbohydrate structure, monosaccharides, okay? So the name carbohydrate, hydrate of carbon, refers to the empirical formula CH2ON, where N is greater than 3, is 3 or greater, N usually between 5 or 6, but can be up to 9. Okay, let's just run through this material. This is 55 pages, so we shouldn't waste too much time. Oligosaccharides are polymers of 2 to about 20 monosaccharide residues. The most common oligosaccharides are disaccharides, which consists of 2 linked monosaccharides. I was talking to you about this. So you have one monosaccharide here, monosaccharides. And let's say they are both glucose, so they link together. Water is lost through the reaction, and they form a disaccharide, and they can form bigger chains after that. So that's what they're talking about. Okay, so let's keep going. So polysaccharides are polymers that contain many, usually more than 20 monosaccharide residues. Glycan is a more generic term for carbohydrate polymers. So say glycans, MCQ question style stuff. So I'm just going to jump through all this stuff because you can go study this on your own. Monosaccharides are water-soluble, white crystalline solids that have sweet taste. They have sweet taste because you can tell like glucose and fructose are examples. They have good and sweet taste. So chemically, monosaccharides are polyhydroxyaldehydes or aldoses or polyhydroxyketoses. So don't let this thing confuse you. They are using plenty big grammar. Poly, you know what poly means? Plenty. Hydroxide. Aldehydes, you know what aldehydes are? Aldehydes are compounds that have the OH group at their terminal carbon. So you have carbon here, OH here. So this is an aldehyde. An aldehyde group is the double bonded carbon at the terminal end. This second one, we already talked about poly. Hydroxy, ketone. Ketone is that they have their hydroxy group not at the terminal end, but this time they have it at the middle carbon. So here, probably the second carbon most likely. So maybe I should put OH here. I'm not sure if I should. Or maybe that's CH3. Okay. So I will check it in a few slides. So this is a ketone. Acetone. OH. Okay. So ketone and aldehydes, don't let all this big language confuse you. They're classified by their types, carbonyl group, and the number of carbon atoms. Okay. The suffix "-ose" is used for naming carbohydrates, although there are a number of exceptions. Of course, there are always exceptions. All monosaccharides contain at least 3 carbon atoms. One of these is the carbonyl carbon, and each of the remaining carbon atoms bears a hydroxyl group. Don't let this confuse you. Simple words. One of these is the carbonyl carbon, and each of the remaining carbons bears a hydroxyl group. So the carbonyl carbon can be either the ketone or the aldehyde. Remember we talked about it just now. The ketone or the aldehyde. So if it's the ketone is this guy, if it's the aldehyde is this guy. So all they're saying is that when there's a ketone or aldehyde, the carbonyl group is the only thing that doesn't have hydroxyl. Everything else will have hydroxyl group attached. Hydroxyl group here, hydroxyl group here, hydroxyl group here. That's all they're trying to say. So Fisher's projection, the most common kind of projection you should need to know. L, just on the left side, when OH is on the left side, that is when OH is on the left side, that is L glyceraldehyde. When OH is on the right side, that is D glyceraldehyde. Let's just speed through. I want to make this video as short as possible so that you can go back and study on your own. This shouldn't be more than 10 minutes at most. The convention of designing D and L isomers was originally based on the optical property of glyceraldehydes. So listen to this. Now they wanted to make a standard sequence of how they did things, but problems arose because after they tried to do the D and L, so in reality, although they named them D and L, there's no real symmetry between the D and L and their stereo optical properties. So in other words, the form of glyceraldehyde that causes rotation to the right is dextrorotatory, was designated D for glyceraldehyde, but it doesn't apply for anything else. So just remember D and L, and remember that I think what's the most common one in this system? D is more common. Of course, in amino acids, it was L that was more common. So L was naturally occurring, but in carbohydrates, it's the D that is naturally occurring. So let's keep going. Okay, so if they ask you a question, how many stereoisomers do aldotetros and azohexos have? So what you need to do for this, to understand this, is that know that this was a typo they made here. This is not 2n, it's 2 raised to the power n. I don't know how your PowerPoint looks like, but it's supposed to be 2 raised to the power n. So that means aldose tetros, 1, 2, 3, 4. Aldose here, double bond OH. This is a H, this is an OH, this is a H, this is an OH, this is a H, H, and OH here. Then aldohexos, 1, 2, 3, 4, 5, 6, right? Are we together? You know what I'm doing, right? I hope you understand why I'm doing what I'm doing. If you don't, please reach out to me and I can explain all of this. But for the sake of your exam tomorrow, just take it as it is. Okay, what a chiral carbon is something that has 4 different groups around it. For example, this, where am I going? This guy does not have the same group around it. It has hydrogen and hydrogen the same, so it can't be chiral. This one does not have 4 groups, so it can't be chiral. This is the carbonyl carbon, and this is the alpha carbon. The first carbon away from the carbonyl carbon is the alpha carbon. This guy cannot be chiral because it has 2 hydrogen ions. So let's count the number of chiral centers, shall we? Okay. So this is chiral. 4 different groups around it, right? And this is chiral. Only 2 chiral centers for aldose tetros. That is 2 raised to the power n, 2 raised to the power 2, that's 4. So aldose tetros has 4 stereoisomers. Aldose hexos, let's count. 1, 2, 3, and this is 4. So what we now have there is 2n, that's 2 raised to the power 4. That's 2, 4, 8, and that's 16. So aldohexose has 16 stereoisomers. Of course, if we go down, we realize that ketose is a bit different. Let me take away these labels I had here. So ketose is a bit different. So ketose is just like 1, 2, 3, 4. So double bond is here. Remember that for ketose. Don't ever forget that. And the only difference between ketose and aldose is that you have 1 less chiral carbon on ketose than aldose. So let me show you what I mean right here. Okay. So with this, what you have is this chiral? No, because hydrogen and hydrogen are here. Is this chiral? No, hydrogen and hydrogen are here. So the terminals are usually not chiral. The terminal ones are usually not chiral. So what else? The only chiral you have? You have only this guy, the only chiral. So there's only 1 chiral center here. So how many stereoisomers you have? 2 raised to the power n. That's 2 raised to the power 1. That is 2. So I hope that answers your questions about this. So I want to be speeding through because I know I don't want to make this video too long. Uploading will also be an issue if it's too long. So we have dihydroxyacetone. Just know them. These ones are the most famous ketoses. D-erythrulose. You guys start using them when you talk about Krebs cycle, citric acid cycle, and everything like that. But for now, we just take them as it is. Cyclization of aldose and ketos. So this, you form a hemiacetal. This is one of the favorite compounds of many researchers, because what this is, you're combining an alcohol with an aldehyde or a ketone, and you end up forming an alcohol on one group and an OH on one group and an OR on the other side. So it's kind of a weird compound because it has two oxygen on either sides. So you can realize that this will have some really weird and amazing properties because of its amount of pull of electrons away from this central carbon. So it's very pertinent to researchers. So just know what it is. A hemiacetal, and hemiacetals are chiral according to this, because there are 4 different groups around them. 4 different groups, 2, 3, 4. Okay. So let's run. So there are rings, and hemiacetals are the intermediates to form the rings. You have the pyrene and the furan. Pyrene is the 6-membered rings with 5 carbons. 1, 2, 3, 4, 5. Then 6 is the oxygen. Then the furan is the 5-membered rings with 4 carbons. 1, 2, 3, 4. Furans are formed by ketoses mostly. Pyrenes are formed by aldoses. At least aldohexose and ketohexose form furans. Okay. So let's just run, right? Shall we? Hawthorne's projections are used. So you have the electron push diagram. You need to not know that too much, but yeah, you can go study that a bit. Or of course, remember the bond angle. The geometry of carbon atoms on a monosaccharide ring is tetrahedral. That's 110 degrees. So you have a boat-shaped conformation. You can have envelope-shaped conformation. Even though I'm running through the slides because I want to make this video as short as possible for you. So you have a chair conformation here, you have a boat conformation here, and I have a Hawthorne's projection here. Yeah, so just realize something. When you ask you which is more stable, the chair conformation and the boat conformation. In real life, which is more stable, a chair or a boat? A chair is usually more stable, so the chair conformation is actually more stable. It has less, the more stable conformation is one, look at it. The most stable conformation is one in which the bulkiest ring substrates are equatorial. So the chair conformation will be equatorial. Equator, that means they're pointing up and down, which means that they're pointing up. Where is it? Up and down, equatorial. In fact, this conformation of beta-D-glucose has least steric strain on aldohexoses. The same question is the most stable of the 2 chair conformers. Of course, it's the one in the equatorial position. Now we need to know which one is equatorial. Is this equatorial or is this equatorial? So let's keep going. Classification. Monosaccharides are those sugars that cannot be hydrolyzed into simple carbohydrates. They are as small as they can be. They are polyabsorbed and have about half the energy use of the cell. Disaccharides, I told you about disaccharides. Oligosaccharides, you know about those. Polysaccharides, they are from 10. They have more than 10 subunits. So just know that the cellulose cannot be digested by the body, just like starch, but we can digest glycogen and stuff in those regards. So glycosidic bonds are very important. They link 2 monomers together. Let's just run. Reducing sugars, monosaccharides and most disaccharides are hemiacetals and therefore can contain a reactive carbonyl group. They are readily oxidized to diverse products, a property often used in their analysis. Just this means is that the hemiacetals are very useful intermediate in all kinds of research. So just know that. I said that before. Glucose, maltose, cellobiose, and lactose are sometimes called reducing sugars. Carbohydrates are acetals, such as sucrose, are not readily oxidized because both anomeric carbon atoms are fixed in a glycosidic linkage. So remember what the sucrose was, right? So you have glucose plus glucose is what? Glucose plus fructose and glucose plus galactose. Lactose minus sucrose. So what they're talking about is carbohydrates that are acetals such as sucrose are not readily oxidized because both anomeric carbon atoms are fixed in glycosidic linkages. Just know that sucrose is not easily oxidized. That's all you need to know. There's so much to talk about in these slides. Like this whole slide could have literally been a whole semester, but gladly they gave us just one slide. Just know this stuff like FAD and NAD are also nucleotides, that they have bonds that are relevant to us. N-glycosides. Just read through stuff. Of course, cellulose and chitin are structural polysaccharides. There are two types, structural and storage. Starch and glycogen are storage, so you should need to know the difference. Storage versus structural. D-glucose is synthesized in all species. So let's run. Amylopectin is a branched version of amylose. Yeah, I can't really explain too much now. But okay, look at this. Branches or polymers side chains are attached via alpha 1,6 linkages and glycosidic bonds to a linear chain of residue link. So what they're trying to say is this: when there's a linkage between carbohydrate molecules like this, you have this, right? So up to 25 residues, and then if you want to link out to a second chain that does the same thing, you need an alpha 1,6. These guys are alpha 1,14 linkages, but alpha 1,6 is what causes the branching. So alpha 1,6 and alpha 1,4 are combined, and look what it says. The side chain contains 15 to 20 glucose residues, and branching occurs on average every 25 residues. So after 25, another branch happens here, and it continues like that. So this is how you get a lot of energy, because you can actually reduce all the ends. You can reduce here, and get 3 monosaccharide units, and in one full sweep, 3 glucose molecules are released, as opposed to if they're all in sequence, one after the other. You will have to take a long time to release all of them. Okay, so just know the numbers. Amylopectin molecules isolated from living cells may contain 300 to 6,000 glucose residues. That's a lot. Know that your body cannot synthesize beta glycosidic bonds. It can only synthesize alpha. And of course, there are 2 types of bonds. Alpha 1,6 bonds are not substitutes for either alpha and beta. Right. Beta amylase is an exogenous exoglycosidase. It acts on terminal glycosidic bonds. What it does is that it breaks terminal glycosidic bonds down. Then alpha, where is alpha? Alpha amylase catalyzes random hydrolysis of the glycosidic bonds. So just know that beta goes to the terminal ends. You know I talked to you about terminal ends. These are the terminal ends. These are the terminal ends here. Terminal ends. But alpha doesn't go to the terminal. Alpha just picks anything in between and just cuts and basically cuts the structure into smaller units that allow for the process to happen all over again. I think we're going to end that for this carbohydrate slide. Up to 10% of the mass of the liver, of course, is stored in glycogen. Yeah, go study, guys. Beta 1,4 linkages are not broken. Just know that they are not broken by our body. Cows and cattle can break it down, but we can. Ruminants can, but we can't. So if they ask you which bonds can be broken down, alpha can be broken down. Alpha is a go. Alpha is a big green yes. And beta is a big red no. So we cannot break down beta. Beta 1,4 glycosidic bonds. And I should tell you why they're alpha and beta, shouldn't I? But you just need to know these values, and just know that one of them, the OH is higher. The OH is higher up here, and for the other one, beta, the OH is on top, but for alpha, the OH is actually at the bottom. So that's how you can separate alpha and beta glycosidics. You can get into the details later on. We can make this thing more complex, but just know that alpha and beta are like this. Okay, so we are going to end this one right here. I hope you guys study well, and I thought we'll get through everything, but at least we did carbohydrates, so I mean, you can study the rest. Don't sleep on nucleotides. Don't think it's too easy. Go study it and get some results. Okay, thank you very much, and I hope this is useful to you. God bless.